That was a mouthful. Well, I'm going to be much more abbreviated at the introductions of my panelists, although they really are deserving of all of your attention. So first of all, thank you for coming. I know we are the last panel standing between you and your lunch, so we're going to be sharing a lot of really exciting news about single-cell genomics analysis and the advancements in the space. And I'm really honored to be joined today by three really spectacular panelists. First to my left is Peter Smibert from the New York Genome Center. And he leads the Technology and Innovation Lab there that's focused primarily on developing tools in genomics. Most recently, they've really focused on single-cell genomics tools and with an emphasis on multiple modalities of information in high-throughput single-cell analysis. So really an expert joining the discussion today. Then there's Charlie Silver. He's the CEO and co-founder of Mission Bio, where he spun the company out of UCSF in 2014. He led the company's evolution from research and services through to the product-based launch, where they are now enjoying a market leadership position in DNA-based single-cell analysis. Mission Bio now has full multimodal omic capabilities, serving applications across pharma and academia. And last but certainly not least, Hanli Ji. He's a medical oncologist and clinical geneticist, serving as a professor at Stanford University. He works on developing new sequencing technologies applied to translational and basic research topics in cancer and other applications. So thank you all for joining today. I'm sure everyone in the audience is seen and is excited and joined today primarily because of the Nature Methods paper, where in 2019 Nature Methods acknowledged single cell as the method of the year. And you know, when single cell analysis first came out in the around 2013, there was promise of the technology, but what was acknowledged in the methods paper in the award of 2019 was the ability that single cell genomics has unlocked around multimodal omic capabilities. And so I wanted to start off the discussion, perhaps with you, Peter, around really inquiring what's the so what? What does multimodal, first of all, it's a tongue twister, so anyone at the end who can say that 3 times fast, multimodal omic measurements, gets a prize. But yeah, can you tell us a little more about what's the so what? Why is that important? What's that giving us today that we couldn't have seen or understood in years past?
Okay, so single-cell omics approaches in the last 5 years or so have done a really good job in letting us see what cells are present in a population. So by looking at the RNA or the DNA copy number or chromatin accessibility by ATAC-seq, for instance, you can get an idea of what sort of cell types are there and whether you have different cellular phenotypes or different cell states. The power of multi-omics is being able to link these, essentially do correlation to correlate different cellular phenotypes to each other. So, examples being correlating the transcriptome with the protein expression, or looking at individual sequence variants and correlating that with cellular phenotype. And I think for now, most of the multi-omic methods so far have focused on a couple of modalities. It's typically I think in the article that Alison mentioned, Michael Eisenstein referred to RNA as being the common currency of single-cell multiomics. And I think for the large part that's true. We're at the moment where we and many other groups are coupling the measurement of RNA with some other modality and sort of using that to infer a phenotype or some kind of a behavior to a cell based on some other characteristic. I think the real power is going to come in the future, where people are, already now working on this, is combining many different omics at the same time. And I guess I'll stop there because I know that the guys to my left have a lot to say on this as well.
That's great.
I think one thing to understand about multiomics in general, and then single cell behind it, is that multiomics is nothing new. It's something that at some level everybody is doing right now to a large extent. Folks are piecing together exomes together with transcriptomes, together with fusion panels, and turning that into an integrated data set that integrates with bioinformatics on the one hand. And then single cell, of course, is the resolution of biology. It's the resolution of complex disease like cancer. Every cell is different, so it's important to study disease at that resolution. And I think single-cell multiomics is really a recognition that once we can finally integrate these data types that we're already acquiring from multiple modalities across multiple platforms and integrate them at the level of the cell where it matters, then you can start to piece together some really important discoveries instead of having to spend all of your bioinformatics stitching together disparate data sets. You get more precise resolution straight off. So on the one hand, it integrates what we're doing already. On the other hand, of course, it enables new discoveries. And I think we see a lot of customers across the market looking at very rare stem cell populations that are the progenitors of disease and understanding based on multiomics how you can identify the cells and then identify the biomarkers that lead to disease progression. And I think that opens up a lot of really interesting applications.
So what would be an example of a killer application? I don't know, Hunley, if you want to field that question.
In terms of this meeting, I think, particularly on the therapeutics development, I see two fronts that are gaining a lot of traction. So one is a preclinical discovery model. So increasingly I hear from my pharmaceutical colleagues who are doing preclinical discovery that this is an informative system to use because it enables deconvolution of cellular complexity. And the more that you look at it, the more disturbing it becomes because much of what was developed in terms of cancer cell lines as an example in therapeutic targets, the readouts that you see are typically very complicated, much more so than was anticipated when some of these things were identified using conventional biochemical bulk methodologies. So I'm anticipating that there's going to be a lot of therapeutics discovery in the preclinical arena will increasingly be informative. Secondly, on the clinical side of things is that you're seeing a lot of traction simultaneously, either for discovery and or for therapeutics identification. So a lot of what's happening in terms of therapeutics within the cancer space is now increasingly a recognition that cellular interactions, whether it's tumor microenvironment or interactions amongst tumor cells or even recognizing the complexity of tumor cells themselves, the type of assays that people have traditionally used to measure those are inadequate because you don't really understand the signal. And perhaps that could explain the failure of IDO1 inhibitors and the billions of dollars that were wasted in those trials. So I think where you're going to see this interaction between discovery, clinical validation is where we start looking at those tumors as an example of an affected disease tissue and understand how these cellular interactions occur. The underlying cellular biology gets deconvoluted. And just to add one thing about it is multiomics is, to me, a code word for something different. Multiomics is a code word for cellular biology, complexity, and the type of readout to use, whether it's biochemical, protein expression, expression-based, gene expression, these types of things. You can summarize that and you can understand that on a more discrete level. You can capture that complexity. And so I'm quite excited about that. I think the next 10 years will be dramatic in terms of the changes we see.
You could almost say multimodal testing is just letting us see what's been there all along, in a way.
Yeah, absolutely.
Yeah.
I think you hit the nail on the head.
How important, and anyone feel free to chime in, how important is it, especially when looking at cancer cells, how important is it to resolve it spatially? And in recognition of the cell-to-cell interaction morphologically, like, is that going to be as meaningful as some blood-based testing, for instance?
Well, there's certainly been a lot of interest. There's all these multiplexed immunohistochemistry panels to look at that. A lot of that's being driven by immunotherapeutics development. Literally every clinical trial I'm starting to see come through where they're testing immunotherapeutics agent asks for a pre and post biopsy of treatment to understand the cellular interactions that are happening. Spatial is two-dimensional. Single cell is three-dimensional. So there are some advantages towards spatial aspects because of the tissue types that are used, but I estimate that the next 5 to 10 years, and maybe Peter's already working on this, I certainly know I'm thinking about things, is that even single cell as it stands now can be used to resolve spatial information. So the type of features that we're looking at regarding cellular heterogeneity, proximity, and distance that indicates biological interactions, increasingly there'll probably be in silico ways or single cell ways that will also be able to highlight those in addition to spatial.
Yeah, I agree with everything Hunley just said. I think as things currently stand at the moment, a lot of the spatial methods probably lack the resolution that we can get by dissociated single cell methods. I know this is changing rapidly. At the moment, I think a lot of the workflow is probably doing very high throughput dissociated single cell assays, multimodal assays, and then computationally integrating that together with spatially resolved technologies. But I think everyone would love to be able to look at these samples in their native contexts and really get an idea of what cell is next to which other cell, and maybe how is that influencing the way that the cell's behaving.
I think one of the questions that's kind of lingering for me is I have two great research centers on the panel, and you guys are really deep into single cell and multimodal. But where is the clinical utility, and how far away is this great science? Is this really great funded science projects? It sounds like, how far away are we from the translation of these tools and whatnot into impacting patients? I don't know, Charlie, if you want to kick us off with your thoughts there.
Yeah, I think where we've built into the space with the DNA platform, we've started to see the leading edge of where clinical utility is going to land, and I think we'll continue to see that over time. DNA is naturally more of a clinical analyte. It's easier to make a robust and quantitative measurement. The sample quality tends to be more robust in terms of handling. And for all of those reasons, that's where you tend to see DNA land in the clinic. Where we've started to see it pull through is in disease areas where the genetics are very well understood. So when you take it into single cell, it's already been laid out by years, or in some cases even decades worth of research with traditional bulk NGS or other genomic assays. So when you bring it into single cell, you know what you're looking at. And then single cell gives you the increased sensitivity that you would otherwise get with very deep sequencing, with the added benefit of understanding how different genetic pathways play together at the level of the cell without averaging those out the way you would do in bulk. So specifically, we've started to see pull in minimal residual disease monitoring, therapy resistance, and understanding therapy response and resistance in myeloid malignancies, where, again, the genetics are very well understood, clonal architecture driving the disease is very well understood, and then single cell is a drop-in replacement that gives you better sensitivity and better confidence in understanding at the single cell level what the disease is actually doing.
What's the so what there? So if my family member has AML or multiple myeloma or something, what's the implication? How could single cell or multimodal testing and looking at being able to resolve cellular heterogeneity, what's the impact then to the patient care?
Yeah, there's always got to be impact, otherwise why are we doing this in the clinic? And I think where single cell comes in specifically for myeloid disorders and some of the others that our customers have started to pull through is, number one, the sensitivity for molecular minimal residual disease. You can identify these very rare cells at low enough levels that you pick them out early in treatment. And number two, once you've identified them, because very digital PCR or other sensitive technologies can do that as well, once you've identified them, you have the cell, and with great confidence that there is a pathogenic cell that you need to be treating. And not only that, within that cell, you understand the resistance genes, so you can start to pick out, okay, this drug is working, this one's not going to, and maybe look at combination therapies, which some of our customers have started playing with as well.
What's the role of cell engineering here?
So, just maybe to tie in the previous question with what you just brought up, is that single-cell DNA has existed for centuries. So karyotyping is typically a single-cell resolution methodology, and it's critical in terms of understanding aspects, say, of AML, where you can consider aspects of aggressiveness, recurrence, likelihood to go on to allogeneic transplant. So now that we can resolve that, I think one of the killer applications, which is science fiction now, but I think will be reality 10 years from now, I'm sure someone's going to do a startup within the next 2 years or so, is that if you understand these complexities about diseases, and let's take allo as allogeneic transplant in the context of AML, could single cell be used to facilitate, accelerate, further refine cellular engineering? So we already do cellular engineering, so there's a whole bunch of people over there talking about CAR T and how we try not to kill people with that methodology. But it's only a matter of time before we can start engineering those lymphocytes using high throughput systems to try to reduce the toxicity up the therapeutic effect. If you had an ability as part of your process, say in a commercial development setting, where you can test that phenotype and understand my toxicity level is going to go down, so I'm not going to give the patients brain edema because they've got lymphocytes going in there causing all kinds of problems because I've turned this off. You could use a single cell format to be able to get through and identify that signal and then identify the drivers or consistent cellular genome engineering edits that you might want to make to optimize that effect. And so the use of single cell to facilitate these types of tissue cellular engineering strategies, I think, is something viable. So let's go into real science fiction. How about an allogeneic transplant or any types of transplant? How about we start to modify the MHC locus to be able to minimize some of those graft versus host things or rejection aspects? Is that science fiction? Yeah, it is now. But as Allison already knows, science fiction is a lot of what happens around here, and you usually get a hit 1 out of every 1,000, and then something spectacular comes out of it. So I think that this paradigm of a cellular engineering system where you use single cell to discretely find your signals that you want to optimize and use that afterwards as the readout that you start building your cellular genome edits to create that cellular construct that does what you want it to do is something that will happen increasingly 10, 20 years around. Hopefully I'm still around when that happens. So
We'll have you back up on the panel in 10 years and fact check. So somebody write it down. Well, I think before we transition, because I want to make sure that we get to contextualizing these tools and these capabilities inside of a market landscape. Before we do that, any thoughts on other killer applications or the so what? Peter, I know you're doing a lot of fascinating work at the New York Genome Center, specifically in this space.
Yeah, I think getting to what Hanli was just talking about, we are doing a lot on using single cell as a readout for CRISPR screens, which may not immediately have clinical application, but in terms of having a very fine, detailed, very fine scale, but also really detailed readout of cellular phenotypes and being able to do these sorts of perturbations in massively parallel manner. I think that we and others are doing this work, and there's a lot of excitement around this because you can get through, you can do the same sorts of assays that would have taken thousands of person hours and years of postdoctoral students' work lives in a matter of weeks now. And so I think just on the tool side, these types of tools will enable people to do these types of assays faster and maybe develop preclinical leads and have impact on patient health that way.
Mm-hmm. Now I think with all the awesome advancements that you each have been speaking about, and I think also the nod from Nature Methods around single cell multimodal measurement, you would think, or at least I have the impression, that it's ubiquitous. At this stage, anyone doing meaningful research in human health is using single-cell multimodal analysis. Is it ubiquitous? And especially as you see the 10x IPO and the great success that they're seeing directly correlated to single-cell RNA analysis, is this something that globally across the world, this single-cell analysis is now the name of the game?
I'd say it's becoming ubiquitous. And Peter, when you and I talked about this earlier, you used a word that I love. I think it's accepted now, which was the word you used, and I think that's exactly the right way to put it. Everybody understands the reason for single cell at this point. Cells are the basis of disease, and you've got to understand information at the resolution of disease to have a chance of understanding it and then fighting it. That's accepted. And I think the last decade or so worth of technologies and science behind it has gotten us there. From there, it's a question of adoption, and the adoption gets us through to a ubiquitous assay that everybody is using. And from there, look, the burden is on us as the tool developers, on you guys as well as the fundamental technology developers, to turn this into something that's just a turnkey workflow that anybody is able to use. Because once you've accepted that you need single cell, information out of it gives you the resolution of disease, then for everybody to be able to use it, we need to turn it into next-gen sequencing today, where you get a quick readout, software is there, and you can make a quick interpretation to get down to the insights you need. And I think that's happening. I think this is the age of single cell. I think it's the age of single cell multiomics, to the point where the solutions plus the software behind it are turning it into something that anybody can quickly pick up and use.
Well, I think one of the things that really supported the rapid adoption of next-gen sequencing was the also decrease in price. And I guess one of the things I'm curious about is, obviously, the bookends here on the panel, you guys must be well-funded, your research is funded. What does it look like from single cell, the utility and the benefits that it's offering as a balance to price? Like, can you speak to what you think the future is of that, and like how the utility of the data that's generated is?
So the costs do come down. I mean, it's obviously there's complex reasons why costs are what they are, but also researchers will find ways to cut corners and save money. If anyone who's done any sort of molecular biology knows that you can dilute kits more than the manufacturer recommends in general. And we and others have done these same types of assays, even building upon the 10x platform. And there's been some really exciting methods that have come out that I'm completely unaffiliated with, that we maybe increase the throughput by about 4-fold, and some of these methods have the ability to increase the throughput maybe 10-fold. So the same outlay of money to 10x gets you 10-fold more cells. You still need to sequence them. I can see Illumina looming over us. Illumina, everything goes through Illumina in the end. But I think the companies will keep producing these great products, but the scientists will also find ways to hack them and play around with them a little bit and add bits here and there.
Solve the price and the software and informatics.
It's a healthy ecosystem.
Yeah, it's a healthy ecosystem. So we have just a couple minutes remaining, and Hunley, I think you were really inspiring me around what you're forecasting into the future with your vision. I asked each of the panelists in advance of the session to imagine I've now placed in their laps a crystal ball, and I want you to share with the audience, from a single cell multimodal measurement perspective, in your crystal ball, what do you think 5 years or 10 years from now we'll be saying about the utility and the adoption and how we'll be using these capabilities 5 to 10 years from now?
I think we will be routinely and relatively cheaply assaying millions of cells in a single assay in a relatively quick format. And what we'll be assaying will be not just limited to the things that we're measuring currently, DNA, RNA, protein, other analytes as well. It'll be very exciting. I wish I knew, but I suspect it's just going to be more things that we're measuring at greater scale and really enabling us to get to even more complexity. And this is assuming that we're also going to have spatial information when we need spatial information. I think that will come.
Look, the reason we're all here, the reason TAL brings all of us together in this wonderful conference every year, the reason we all do this is to make sick people better and to keep the rest of us healthy. And I am eternally optimistic that once we finally have the tools to be able to see the disease at the resolution that matters, we have a shot at actually delivering that. I think the success of the genomics wave over the last decade has been proven out in cases like Gleevec and Herceptin and drugs that result in thousands, hundreds of thousands of people still walking the earth today. They're alive because of those drugs. But at some level, it's still very much blind people groping in the dark for the best solution. But I think once we finally have the tools at hand to be able to read out every piece of information that disease has to tell us at the resolution of disease, I'm eternally optimistic that over the next 5 to 10 years, we're going to knock out diseases one by one.
I am going to anticipate a couple of things. So single cell actually is more than single cell. Single cell is really reducing biology in any context, disease or otherwise, to cellular quanta into a digital format. So where we look at the Googles of the world or Facebook or Amazon where you buy cheap junk, the difference with that, though, is the context is direct. It has a direct impact on our lives. And the ability to digitize the function and processes of biology, whether it be disease state or anything else, at this level of this fundamental quanta, cellular biology, that will lead to a whole revolution where we once thought about aspects of how compute and cloud computing brought about. This is the digitization of all of biological sciences. And so the kinds of things and developments you'll see based upon this complexity that we can now do better jobs of interpreting, as well as integrated within our ability to manage complex data sets and draw out inferences, I think there'll be astonishing things that come out of that.
Well, it's very inspiring. Well, join me in thanking the panel and have a great lunch.